Hello, and welcome to my seminar titled Measure Once, Cut Twice, How CRISPR-Cas is Revolutionizing Biotechnology. The objectives of this seminar are to discuss what is CRISPR-Cas, how it works, why is it so powerful, and what are its many applications within biology, biotech, and medicine. At the heart of this discussion is genetic variation. Genetic variation is the difference in DNA sequences between individuals within a population. This forms the basis for evolution, our individuality, as well as diseases such as cancer. Variation can occur in germ cells, which are passed on to our offspring, or somatic cells, which make up every other cell within our body. Mutations and recombination events are the major sources of variation within genomes. Genome editing is a process by which DNA is inserted, deleted, or replaced using nucleases to modify the genome of interest. These nucleases cause DNA damage, which are repaired by the cell, resulting in mutations being incorporated. It's important to repair DNA damage. If the DNA damage is left unrepaired, this can result in death of the cell. Double-strand breaks, which are typically caused by the nucleases, are repaired by two general mechanisms. Non-homologous end joining, where the DNA is ligated back together. This often results in inserts and deletes extra nucleotides at the site of repair, resulting in mutations. If this occurs within a protein coding region or a regulatory region, this can change protein function or expression of the gene of interest. Double-strand breaks can also be repaired through homologous recombination. This is an error-free form of repair in which a template DNA is used to repair the DNA. If this is the other gene, this can be error-free repair, or if it's a template DNA provided to the cell, this can incorporate a desired mutation at the site of DNA damage. Historically, we have used non-specific endonucleases to cause a DNA damage. These nucleases are fused to DNA binding domains, which can be generated to target specific regions within the genome. However, this is labor-intensive and costly. Cas9 has changed all this. CRISPR and Cas9 were identified as a form of adaptive immunity within bacteria. When a viral protein infects a bacterial cell and injects its DNA, this foreign DNA can be identified by Cas proteins, resulting in its degradation and the incorporation of a small piece of this viral genome within the genome of the bacteria in the form of a CRISPR array. Once this CRISPR array is expressed and matured and processed, this CRISPR RNA can be loaded into Cas proteins such as Cas9 and be used to identify any subsequent foreign infection by viruses and foreign DNA. That foreign DNA is degraded and infection is prevented. If we take a closer look at Cas9, we see that it's a multi-subunit nuclease with two nuclease domains. When it's loaded to a mature guide RNA, it's able to identify target DNA, resulting in a double-strand break. A key aspect of Cas9 is not necessarily the ability to induce a double-strand break. It's the ability to be programmed to target any DNA within any genome. By providing a guide RNA, we can target the Cas9 to any point in any piece of DNA in any genome. Mutations within a nuclease domain can also be used to generate different forms or different variants of Cas9. Mutating a single nuclease domain generates a nickase, which can be used to increase the specificity and reduce off-target effects of DNA damage. We can also mutate both nuclease domains, resulting in a dead Cas9, which is unable to cause any damage to DNA, but is able to be used as a shuttle to bring effector proteins to specific regions within DNA. These effector proteins can influence transcription, epigenetic regulation, or even label DNA in specific sites. New forms of Cas9 with different effector molecules are being generated constantly to allow researchers to study different mechanisms of biology. CRISPR has revolutionized genome editing because it's cheap, it's efficient, and it's fast. The analogy I like to use when comparing CRISPR to talons is that of a self-driving car. With talons, we had a self-driving car that could only take you to one place. If you wanted to go someplace else, you needed to rebuild the entire car. With CRISPR, we now have a self-driving car with a GPS. We can program it to go wherever we want. This leads to its many uses and applications within biology. As a research tool, we can use it to generate knockout models within cell lines to study the function of specific genes. In this case, Cas9 with a guide RNA can be used to generate a random mutation or a specific mutation within a cell line of interest in order to study the function of a particular protein. I use this in my work to study the regulation of the MYC oncogene. By knocking out regulators in the MYC oncogene, I can study their effects on protein function. If we don't know the causative gene for a phenotype of interest, we can use a genetic screen to identify potential genes of interest regulating or causing a specific phenotype. In this case, we use a library of guides targeting every gene within a genome or a subset of genes, packaged into a virus, and used to infect cells. By enriching for a specific phenotype, and sequencing those cells to identify the guides present within those cells, we can identify potential genes causing that phenotype of interest. This has been used in many applications to study genes important for cancer, involved in therapy and resistance to therapies, as well as causative genes for specific genetic diseases. In the case I'm highlighting here, a genome-wide CRISPR screen was used to identify genes important for HIV infection within cells. In this case, 5 factors important for HIV infection within the host were identified, focusing on the sulfonation pathway and its importance for HIV infection. CRISPR is also important for the ability to generate animal models to study biology. This is especially key when we're looking at being able to translate work done at the bench into the clinic. Without a proper animal model, it's difficult to understand our biology in a more complex organism. In order to generate an animal model, we use similar techniques to in vitro fertilization, where we do a pronuclear injection into the mouse embryo. In this case, we inject Cas9 and a guide RNA targeting our gene of interest. The embryo is implanted into a surrogate mother, and a litter is allowed to form. That litter of pups is then sequenced and genotyped to identify mice carrying our allele of interest, and then expanded to generate a colony of animal models suitable for our needs. Historically, this has been done with embryonic stem cells. However, this is takes quite a bit of time, upwards of 18 months. Whereas with CRISPR-Cas9, we can do this in as little as 6 months. One of the most immediate utilities of CRISPR and genome editing may be in agriculture. Plant geneticists typically use random mutagenesis to generate seeds containing potential mutations which may be beneficial, which are allowed to grow and then identified as carrying ideal traits and characteristics. Plant geneticists have also used transgenic models in which foreign DNA is injected into the genome of these plants, carrying genes that might be important for drought tolerance or resistance to pests. However, these are classified as genetically modified organisms because they contain DNA from unrelated organisms. CRISPR changes all this. CRISPR modifies the genome of the plant itself to provide traits which may induce resistance to pests or drought tolerance, reducing the need for pesticides and fertilizers. Because they don't contain any foreign DNA, they are not considered a genetically modified organism. The case I'm highlighting here is a study looking at the wheat genome and using CRISPR to generate knockouts of 4 alleles important for resistance to powdery mildew, again highlighting its potential utility within agriculture. Finally, in medicine, this is what gets a lot of the headlines in the news. there is a potential avenue for using CRISPR as a therapeutic and changing the way we look at gene therapy. Two main methods of gene therapy can be highlighted. One is ex vivo, in which cells are removed from the body, edited using CRISPR-Cas9, and then re-injected into the body to target and treat a potential disease. Or in vivo, in where Cas9 with guide RNA are packaged into a delivery vehicle such as a nanoparticle and injected directly into the body. There are currently 14 clinical trials using CRISPR-Cas9 to target diseases of interest. 11 are in cancer, typically using a form of immunotherapy where the T cells are removed from the body, edited in order to target the tumor efficiently, expanded and re-injected into the cell, targeting the tumor. There's also clinical trials initiated to target genetic diseases such as sickle cell disease or infectious diseases such as HIV and norovirus. What we can conclude is that CRISPR-Cas has revolutionized the way we look at genome editing because the programmable nature allows us to quickly, efficiently, and cost-effectively target genes and induce mutations in desired genes of interest. Because it's cheaper and it's more cost-effective, its uptake and utility is widespread and has many applications within both biology, biotechnology, and tellingly in medicine. I want to thank you, and if you have any questions, please feel free to contact me at the email below.